So this talk is about a functional biochemical analysis of tumor cell metabolism based on a tracer technology itself that is able to trace or follow metabolic changes that are introduced to tumor cells based on various treatments. I have heard about deuterium depleted water from a friend of mine in Los Angeles who was interested in finding out what is the underlying biochemical pathways that are altered by deuterium depleted water. So then I got in touch with Gabor and obtained the appropriate specimen or deuterium depleted water for the in vitro type of experiments that I'm going to talk about this afternoon. So the main goal of the study was to find out how these tumor cell lines, including a pancreatic cancer, adenocarcinoma cell line, lung cancer cell line, and a breast cancer cell line, would alter their metabolic fingerprints or imprints in response to deuterium depletion in their culture media. And the tracer that we used is a glucose molecule that has 13C substitutions in the first and second carbon positions, and I'm going to show you what they are used for. So we measured what we call metabolic fluxes or exchanges of carbons between glucose and various other metabolites in the cells that include a whole list of intermediates as well as products that I'm going to talk about in more details. So the main aim was to find out what is the underlying mechanism of altering cell proliferation of in vitro grown tumor cells. And we were keeping in mind that there may be certain markers of toxicity and how selective the deuterium depleted water is on various biochemical reactions, as well as how efficacious this particular treatment modality is. So the approach we took was we actually restored culture medium powder in various concentration, in various strengths of deuterium depletion, and we looked at from the culture medium glucose lactate, carbon dioxide, and glutamate as the product of glucose metabolism. And from the cell pellets, after incubating the cells for 72 hours, we looked at various fatty acids and their synthesis patterns, including myristate, palmitate, stearate, oleic acid, arachidic acid, as well as we determined what is the contribution of glucose-derived carbons to fatty acid synthesis, as well as RNA ribose synthesis patterns in the pentose cycle. And there are many details of these synthesis patterns that can be mapped by a tracer glucose substrate, which I'm going to show you in short. But anyways, it's really truly the main idea is just finding out what is the metabolic adaptation or what is the metabolic response of these tumor cells to deuterium depletion, and the markers or the biological endpoints included glucose uptake from the culture media by these tumor cells, lactate production from glucose. We do know that tumor cells are sugar junkies, and they actually utilize and use glucose very intensely for survival as well as for proliferation. We were interested in how much and what fraction of that glucose gets completely oxidized in the TC cycle, which is the CO2 product of the glucose tracer. We were interested in finding out how TC cycle anaperiodic flux is changing in response to deuterium depletion. Anaperiodic flux means the anabolic use of glucose. So any glucose that is not disposed, or any substrate that is not disposed in the form of 13CO2, then it's used for some kind of an exchange or net production of TCA cycle metabolites or intermediates. We're interested in glycogen synthesis, de novo fatty acid synthesis, their elongation, desaturation, and acetyl coenzyme A contribution from glucose and the pentose cycle. So these studies are fairly robust as far as restoring the metabolic map that is behind any physiological change, if there is any. And I have to tell you, I'm the skeptic scientist kind, so I was not expecting to find any major change in the metabolic network. In deuterium depleted media, in fact, there were. And to my surprise, some of them were very evident, clear, and were very much in line with some mechanistic studies that we did with certain pharmaceutical drugs in the cancer area of primary kinase inhibitors, and truly there was some functional and mechanistic understanding that we could, at least in these three cell lines, we could report back to Gabor, as well as we presented them at various meetings. So the method that we used were practically the usual scenario where we separate the media and cell pellets, we extract these metabolites, which I described you a few minutes ago, and chemically derivatize these metabolites, and then we use gas chromatography and mass spectrometry to determine the positional enrichment with 13C in the products. We use the chemical ionization as well as electron impact ionization in some metabolite analysis. When we we were selectively monitoring certain ions of these target metabolite clusters, and then we determined the mass isotopic distribution in these products of glucose metabolism. Our primary interest is to find out, and this is the scheme that shows you how the technology works. We were primarily interested in how glucose entry or how glucose intake would change in response to deuterium depletion, and how specific reactions related to glycolysis as well as production of certain intermediates and products, for example, fatty acids and complete substrate oxidation as well as glycogen turnover. What is the effect of deuterium depletion on intermediary metabolism? In particular, we were interested in what is the effect of deuterium depletion on the oxidative branch of the pentose cycle and the non-oxidative branch of the pentose cycle. Now, the oxidative branch of the pentose cycle is particularly interesting because this is the chain of reactions or the committed steps for nucleic acid ribose synthesis. And in the meantime, while glucose is oxidized directly on the first carbon, there is NADPH produced, which is the reducing equivalent and the source and the strength for de novo fatty acid synthesis and cholesterol synthesis. Interestingly, this particular chain of reactions uses water as the source of hydrogen. So we were looking at these fluxes with particular interest. And then following down the line, the fate of glucose, we were obviously interested in various other products, including lactate and citric acid cycle or Krebs-Seniory cycle metabolites, as well as fatty acid synthesis. Labeling in glycogen from glucose can be through direct pathways as well as through gluconeogenesis. So glycogen analysis would allow us to determine many reversible as well as the irreversible reactions or the side passing reactions of glycolysis. Particularly, there are PEP-CK activities and the fructose biphosphate cleaving activities that are actually irreversible processes in gluconeogenesis, yet we were looking at glycogen just to find out how overall metabolism of these tumor cells is affected. Just to give you a highlight of how the technology works, throughout the schemes there is going to be these red carbons which are 13C or heavy carbons of glucose.